So now look at this action potential. Action potential. So action potential basically is not a particular potential. Like you look at this resting potential, we know it's negative 65 millivolts, right? So you look at this resting potential, we always know negative 65 millivolts. But action potential is not a single membrane potential, but it's a rapid change in the polarity across the axon's membrane as the nerve impulse occur. Okay, so action potential will have these three phases. Okay, three phases. Depolarization, which can be subdivided into slight depolarization and continue with the rapid depolarization. Okay, so first we know that at resting membrane potential or resting potential, the membrane is polarized. So now what we're going to do, we want to depolarize the membrane. So from negative 65, we make it become less negative and eventually become positive. Are you clear? So it starts from negative 65 and then we make the remove, D mean remove. So remove the polarity of the membrane. So it's no longer polarized and from negatively charged become positive. Okay. Then after that, immediately when reach a certain value, we were going to have the repolarization. Are you clear? Basically, we switch it off and then we switch it on again, something like this. Okay, so that we have a repolarization. So re means that we want to redo. Okay, make it become polarized. And then sometimes, most of times, we do have a short hyperpolarization. So it means that after we polarize, we overshoot a bit. Right? We overshoot a bit. Okay, so this action potential, normally we use this voltage gated ion channel, a bit special ion channel here, highlights voltage gated ion channel. So which voltage gated ion channel means that this sensitive. Okay, sensitive to the change of membrane potential. So means that this voltage gated ion channel, usually they are closed unless we do have a certain voltage or certain membrane potential, then they will open. Are you clear? So this one we name it as the voltage gated ion channel. So what they do, they change the permeability of the plasma membrane to sodium ions and potassium ions. So we have two different types of the ion channel, the voltage gated ion channel. One is the voltage gated sodium ion channel. Another one is the voltage gated potassium ion channel. And because of this voltage gated ion channel, then we will be able to have this action potential. Okay. So now let us look at this diagram first. Okay, we look at this diagram. Now, if we look at the entire events, I'm going to do a summary, a quick one here. A bit confusing, so please do follow. Any question, please do let me know. So first, we maintain a resting potential. So at the phase number one. Phase number one, resting potential. So during resting potential, you can see that. So voltage gated sodium ion channel, voltage gated sodium ion channel, voltage gated potassium ion channel, they are closed. So means that sodium ion cannot move in, potassium ion cannot move out. Are you clear? Can you see that? Sodium ion cannot move in, potassium ion cannot move out. So how we maintain the resting potential? We maintain the resting potential by using two mechanisms. One is the active transport, okay, of the sodium potassium pump. Second, membrane permeability. So with this membrane permeability means that we will be able to have more potassium ion leak out compared to the sodium ions that move in. Okay. So we say that at this resting membrane potential or resting potential, we do know that the membrane is polarized. So the first one, at the resting potential, our membrane is polarized. Okay. So what happens to the second phase, depolarization. So here write down is a slight depolarization. Okay, slight depolarization. So what actually happens means that we are going to have the stimulus. Let's say someone touch you. Okay, so when someone touch you, so means that something happened and make it become depolarized, remove the polarization. But this process takes slow period of time. Can you see that? The gradient is not so steep. So it gradually increase from -65 become -64 and so on. And because there is a change of the membrane potential, correct? Slight change, right? When this slight change take place, you can see that some of the voltage-gated sodium ion channel, some of this voltage-gated sodium ion channel open. And because outside more sodium ion, inside lack of sodium ion, so sodium ion move in. But can you see that voltage-gated potassium ion channel remain closed? So means that only positive ion, sodium ion can move in. So when sodium ion move in the cytoplasm, it become less negative lah, correct not? Increase in the membrane potential. Why? Because become less negative. Sodium ion coming in, balance one negative charge ion. Another sodium ion balance. So means that you can see there is an increase in the membrane potential from -65 to -64 and continue until they reach a value called threshold value. What's mean threshold value? Threshold value is very important to make sure that we must reach this threshold value, then only we are going to proceed into the second phase or third phase, sorry, in the third phase termed as a rapid depolarization. Okay, it's termed as rapid depolarization. Okay, so what actually happened? Now look at this. During the phase 2, slight depolarization, some voltage-gated sodium ion channel open. But when you reach the threshold value of -50 millivolts, okay, -50, because why? Because sodium ion keep on entering. When the sodium ion enter, what will happen here is the voltage from -65, okay, let's say one enter, -64, -63, -62. Sodium ion enter until we get -50. Once it get -50, you can see that all voltage-gated sodium ion channel open. So when have all voltage-gated sodium ion channel open, means that there is a massive rapid influx or a fast and rapid influx of the sodium ion. So many sodium ion enter into the cytoplasm. So you can see that now it will have immediate increase of the membrane potential from -50, -49 and shoot up to +40. Can you see that? It shoot up. So means that depolarization, we remove the polarization and continue to make the membrane potential from negative become positive. Then action potential is generated. Because why? It's something like a light. If I switch off the light, there's no signal. But when I switch on the light and then remove, switch on the light, switch on the light. Can you see that? There is a signal. Can you see that? There is a signal. Switch on the blue color point. Can you see that? Switch on, switch on, switch on. Okay. So what actually happened here is you manage to turn the membrane potential from negative to positive. Is considered now action potential is generated. So with the generation of this action potential, when reached to the +40, action potential is generated. But at +40, immediately voltage-gated sodium ion channel, all of them close immediately. So means that sodium ion cannot enter. So maximum until +40, because no more sodium ion can enter. When no more sodium ion can enter, then we maintain at +40. But at the same time, can you see that voltage-gated potassium ion channel open? When the voltage-gated potassium ion channel open, potassium ion now move out. When the potassium ion move out, inside of the cell now lack of positive ion, lose the positive ion. So means that from positive now drop to negative. So means from +40 drop all the way until negative 65. Can you see that? Why? Because we lose the positive ion, potassium ion out. So with this phase is known as the re-polarization. Are you clear? So we write down this term as re-polarization. Why? We make the membrane now turn back to negative 65 millivolts. Okay. So when it dropped to negative 65, then you can see that by rights, voltage gated potassium ion channel have to be closed. Voltage gated potassium ion channel have to be closed, but the problem here is they close slowly. So when they close slowly, it means that there are some extra potassium ion move out. There are extra potassium ion move out. So when there are extra potassium ion move out, so we have undershoot. Because we lost more potassium ion, you have a little bit of undershoot here. So this undershoot is termed as hyperpolarization. 2 over. So until this process will until potassium ion channel or voltage gated potassium ion channel close. When this potassium ion channel close already, then we maintain the maintains the membrane potential back to the resting potential. So the whole series of events here is considered one action potential. Okay, consider one action potential. Are you clear? Okay, so phase 1, resting potential. Phase 2, slight depolarization. Phase 3, rapid depolarization. Stage number 4, repolarization. Stage number 5, hyperpolarization. So with the stimulus. So what stimulus will do to this? Stimulus actually will cause the increase in this membrane potential. Okay. So let us start now. So this is a neuron. So example of this neuron is a Pacinian corpuscle, sensitive to the touch and the pressure. Okay, so as usual, we do know that they have this sodium potassium pump. Okay, sodium potassium pump. And we do know that the membrane is more permeable to the potassium ions compared to the sodium ion. So potassium ions, blue color. So this blue color indicates the potassium ion channel, just a normal potassium ion channel. Okay, with the green color here indicates the, sodium ion channel. So because of these two now helps to maintain the membrane's resting potential. So if I draw a graph now, so membrane potential, millivolts versus the time. Okay, I need longer, sorry. So time. Okay, and because of these two mechanisms, the sodium potassium pump and the membrane permeability, more permeable to the potassium ions compared to the sodium ions. Therefore, we maintain the membrane potential at negative 65 millivolts. Can you see that? No stimulus. So this one, I've termed it as phase 1. So phase 1 is the membrane resting potential. Okay, it's a resting potential. Okay, so in this case, membrane is polarized. Okay, membrane is polarized, no stimulus. Okay, so maintain at negative 65 millivolts. Okay, so what happened now is, if you look at this Pasinian corpuscle, they have this what we call the capsule. Okay, this is a capsule. And under this capsule actually covers the ion channel. Okay, what kind of ion channel? It covers the sodium ion channel. Okay, it covers this sodium ion channel. So means that without stimulus, what will happen? Without stimulus, you can see that it covers sodium ion can enter or not? Cannot, because we have to understand that because of sodium potassium pump, concentrations of the sodium ions is very high outside compared to the concentrations of the sodium ions inside. It's low. How about potassium ion? Potassium ion concentration is low outside, but inside you have potassium ions with a high concentration. So sodium ion direction will move from outside into the cells, correct? If let's say we open it. Okay, so again, the green color here indicates that the voltage, the normal sodium ion channel. Okay, and then next, we do have some special ion channel. These ion channels, they are potassium ion channel, but can you see that it's gated? And also the sodium ion channel is also gated. So now look at this. This one we term it as the voltage gated sodium ion channel. And the blue color is the voltage gated potassium ion channel. Both of them are sensitive to the change of the voltage. Okay? Both of them, they are sensitive to the change of the voltage. Okay, so how the stimulus actually change the voltage? So try to imagine when we touch on this capsule. When we touch on this capsule, the capsule deform. Okay, we touch it out of shape, right? When you touch it or press it out of shape. So when it's out of shape, what will happen here? Sodium ions now can enter. Correct. Sodium ions now can enter into the neuron. Why enter into the neuron? Because outside sodium ions high concentration, inside sodium ion low concentration. So sodium ion now enter into the neuron. So means that inside now have extra positive ion, balance the negatively charged ions. So once we have this action potential, what will happen here is you can see that now membrane potential increase from negative 65 to negative 64. Can you see that? That is a slight change in the membrane potential. And this slight change of membrane potential will cause some of these voltage gated, okay, sodium ion channel to open. Can you see that? Some of them open now. So because of openings of the voltage gated sodium ion channel, so more sodium ions now enter. Can you see that? More sodium ions now can enter. So with the entry of the sodium ions, what will happen here is the membrane potential continue to increase until what? Increase until negative 50 millivolts, which known as the threshold value. So take times until. So this is phase number 2. So what actually have phase number 2? Phase number 2 we call as a slight depolarization. Characterized by what? Characterized by the some voltage gated sodium ion channel open, but voltage gated potassium ion channel remain closed. Are you clear? Voltage gated potassium ion channel remain closed. Okay, some of them only, not all of them open, some of them open, but you can see that if they manage to reach a negative 50, which is the threshold value, okay, or threshold potential, Now what will happen? Then it will enter into the phase 3. What is the phase 3? Now you can see that all voltage gated voltage gated sodium ion channel open. So means that if all of them open or majority of them open, so means that the sodium ion now have a massive or rapid influx of the sodium ion. So more and more sodium ion now enter. Why the sodium ion can enter? Because outside again very high, inside very low concentration of sodium ion. So facilitate diffusion take place. Then we can see that immediately with a short period of time, the membrane potential now shoot up to positive 40. So this is phase number 3. So what is the characteristic of phase number 3? Phase number 3, we term it as the rapid depolarization. Characterized by what? Or voltage gated sodium ion channel open. But how about voltage gated potassium ion channel? Remain closed. Are you clear? Remain closed. So because of the positive ions continue to enter, so we try to imagine from negative 50 become negative 40, negative 30, negative 20, negative 10, 0, 10, 20, 30, 40. When immediately reach this. Can you see that? Okay, immediately reach this positive 40 millivolts, then you can see that all voltage gated sodium ion channel now immediately close. And voltage gated potassium ion channel now open. Are you clear? So immediately now at positive 40, so now sodium ion cannot enter anymore. Why? Because the voltage gated sodium ion channel now all of them close. So at the same time, all voltage gated potassium ion channel now open. Can you see that? When it open, because inside potassium ion channel concentration as a potassium ion actually concentration is higher. Outside concentration of potassium ion is lower. So K plus now move out. So the rapid movement of the K plus from the cells, the neuron, inside neuron, out of the neurons actually decreases the membrane potential. Okay, decreases the membrane potential. So this is what we call stage number 4. So stage number 4 is termed as re-polarization. It's termed as re-polarization. So characterized by what? Characterized by O, voltage gated potassium channel open, and O, voltage gated sodium ion channel close. So because of this, you can see that potassium ion move out, right? So when the potassium ion move out means that inside become less and less positive. So from positive 40 drop to positive 30, 20, 10, 0, negative, and continue all the way until negative 65. Okay, so what actually happened at negative 65? At negative 65 here, so the voltage gated potassium ion channel close slowly. It's not immediately, it close slowly. So when it close slowly, it means that some of the potassium ions still can leak out, right? So you can have a small undershoot. So this is phase number 5. And then slowly you can see that our sodium potassium pump and also membrane permeability bring this back to resting potential again. So this consider one complete action potential. So what actually have number 5? Number 5 we call it as a hyperpolarization. So what actually happened here is during this period of time, voltage gated potassium ion channels start to close, but they close slowly. What happened to the voltage gated sodium ion channel? So remain close. Are you clear? So therefore, you can see that there is extra leak out of the positive ions, therefore making the membrane potential lower than negative 65 millivolts. Okay, so if you need to copy, you can. How I'm going to do this? Okay, so you can take a photo. This is a summary of the process. Let me clean out some of the here. Okay, so I stop the recording now.